Welcome to the Institute of Science and Technology's AI Special Interest Group online seminars, introduced by me, Richard Saldana. These seminars aim to explore a wide range of AI-related topics with invited experts, everything from AI explainability and experimental methods to mathematical challenges. It gives me great pleasure to introduce Professor Francesca Cordeiro, a clinician-scientist, chair and professor of ophthalmology at Imperial College London, honorary consultant ophthalmologist and research lead at the Western Eye Hospital, Imperial College Healthcare NHS Trust, and director of the Imperial College Ophthalmology Research Group Clinical Trials Research Unit. Francesca is also Professor of Glaucoma and Retinal Neurodegeneration at the Institute of Ophthalmology, University College London, and Chair and CEO of recent medtech Novai. She is a trustee for the charity Fight for Sight. Francesca has kindly agreed to speak to us today about the development of DARC, detection of Apoptosing Retinal Cells, a novel retinal imaging technology with many spin-off benefits. This is the technology that is being developed within Novai. Francesca.
So the name Novai actually comes from Nova, meaning enlightenment, and artificial intelligence. And of course, every company nowadays has AI somewhere in the title. But we, and I'm hoping I will show you, use it as integral to our technology, which is a biologic with AI. And I think the whole need for this technology comes from my own experience as a clinician, as a clinical ophthalmologist. And a lot of you will be aware that in ophthalmology, like a lot of other areas in medicine, we are seeing an increased number of aging diseases. And this unstoppable rise in eye and brain disease is really extremely worrying. So you're talking about, for example, every 3 to 4 seconds, someone loses their sight due to age-related eye disease. And that is quite a catastrophic statistic. So 600 million and over people are believed to be affected by blinding diseases by 2050. I think the other one that is interesting, there's the neurological diseases, the diseases such as Alzheimer's and Parkinson's and MS, for example. That's even more, 750 million. The problem is as the disease becomes more advanced, it becomes more expensive to treat. And the argument must mean, how can we overcome this real epidemic of aging diseases? I like using this analogy. I'm not a gardener, but I can assure you that this is quite a good analogy in terms of thinking of a rose patch where the roses are single cells, recognizing the fact that unfortunately, by the time we identify diseases, usually it's too late. So in a disease such as glaucoma or macular degeneration, often the patient presents well after the disease has started. So they've already lost their vision. This is what I mean when I say functional vision loss. Sometimes it's as much as 10 years between when the process starts at a single cell level and when we detect it ourselves with our conventional tests. The structural tests, and a lot of you will be familiar with the OCT tests that you're offered when you go to see your optometrist now. Those are a little bit better than functional changes because those are structural, the tissue shrinkage. But it still is a long time after that first process has started. And so what Novai has done is it's created this AI-powered biomarker that can detect diseases such as blindness and neurological changes at the earliest possible stage. And that is at the single cell level. And we are, at the moment, the only technology that can claim this. So I want to tell you a little bit of the background as to how this all came about. And I think it's useful to highlight that in cell biology, in tissue culture labs all over the world, there is a protein that has been used for many years to identify cells undergoing this process of cell suicide or apoptosis. It is based on the fact that this protein called annexin binds to the cell membrane only when that cell is under stress. Now, we took advantage of the fact that because the eye is transparent, you're looking out at the moment because your eye is transparent, we can use exactly that same axis to look into your eye and, in fact, almost look at your own tissue plate of cells, of neuronal cells. And what we see here is a patient's eye. This is the back of a patient's eye. And you can see these white spots appearing. And each of these spots is a single cell. And what it's telling you is that this patient has some element of disease activity, which it could be at the very early stage of detection before they even go down that pathway of vision loss. So the AI, when I say is integral, it's doing several things. On a very simplistic level, if you look at these red squares, it's counting and identifying automatically these white spots, these fluorescent spots, which is what our biologic is labeling. It's also looking at the distribution of the spots, the density, and very importantly, because the actual biologic is agnostic to what type of cell it binds to, we can look at the actual cell signal to figure out whether or not we can differentiate the cells and also therefore package it into different diseases. So that is actually just one product that has multiple uses in different diseases. So I think what is confusing is the fact that it is one product. We're using this biomarker, and we're currently already working with pharma companies. We're getting revenue from those pharma companies because what we're doing is we're providing a readout much earlier than is currently possible. So compared to a normal clinical trial, which can probably take, 2 years before you know whether or not your drug is working, we can take weeks. We can give you an answer to see whether your drug is working or not much sooner. And that is not only de-risking, but it is also in this world of, knowing that trials are very expensive, you'd like to fail fast if your drug wasn't good. And that's a huge attribute. Where we are going next is using exactly the same good points of the biomarker, the fact that we can identify patients who are at risk of future disease and progressing fast, but also get a very quick turnaround as to whether they've responded to treatment. We want to put that away just from clinical trials into clinic, where it will be used by clinicians like myself as a clinical decision support tool. But ultimately, my vision is for this to be the screening biomarker of choice, to be used all around the world as a way of assessing whether there is any abnormality present, either in the eye or in the brain, but more so to then guide where that person, if they have an abnormal signal, needs to go next, which speciality, and how can they be prevented from losing their memory, losing their vision. So Novai was formed in 2020, and it really came off the back of over £4 million from the Wellcome Trust, which took it from bench to phase 2 clinical trials, and has been making steady progress all the way. Currently, we have had about 400 patients who've received DARC. It's all been extremely safe, very short half-life, and we've had some very encouraging results. As I said to you at the beginning, it is cell agnostic, and actually the AI allows us to focus in on what disease we're talking about. So our lead indication at the moment is macular atrophy. And that's because over the last year and a bit, for the first time ever, there have been 2 new treatments that have come in that can prevent people going blind from this disease. In addition to that form of advanced macular degeneration, and that's called macular atrophy, you also have wet AMD. So a lot of you will know relatives who have injections into the eye. those injections into the eye are specifically for the disease, wet macular degeneration, usually. You can also use diabetes. That will be where we next see us going. And then as we evolve, we'll be in more and more diseases, glaucoma, right down to the neurodegenerative diseases of MS, Parkinson's, and Alzheimer's. So let me go back to macular atrophy. So what is macular atrophy? It is an advanced stage of age-related macular degeneration, AMD. And in that disease, basically you have atrophy, death of the retinal cells. Now, I'll show you a true patient here. This has been published of a 78-year-old lady who took part in our phase 2 clinical trial of DARC. At the time, she had very good visual acuity. So the macula is indicated here by this cross. If you think of your pinhole camera, the way the eye is designed is all the rays come through your pupil to focus on a sharp point of vision called the macula. So if your macula is intact, your central vision is intact. And so this lady had very good vision, despite the fact that she did have this area in yellow of atrophy at the back of her eye. 35 months later, that atrophy had grown. That atrophy is now seen by this red border here. And as you can see, it's now involving the macula, and she has lost her vision. So where DARC comes off its own is at the beginning, it predicted that that lady's atrophy was going to grow and grow in the direction of the macula. And therefore, had this been used to identify that, she could have had her vision saved. We could have prevented her losing her vision. And, you know, there is nothing like that is available at the moment, conventionally, to predict. So this is a good example of why we're going out into these diseases. And being a clinical decision support tool, what I mean is it can identify those patients who are at greatest risk of developing vision loss. So you can identify those who are at high, moderate, and low risk. And what that means, instead of those patients who are at low risk coming repeatedly to clinic so that they can have assessment with conventional measures, you can safely say those at low risk can be seen annually, whereas those that are at high risk and probably will need treatment can be treated early, their vision can be saved, and also they can be intensively monitored. So this is how we would integrate into the clinical pathway to save and preserve the vision. I think this is a bit of a complicated slide, but really what I'm trying to say is that the way we are looking at our go-to-market is in macular atrophy with the instruments that are available, and trying to go out initially with a small team looking at how we can integrate into clinic. But as we move forward, this becomes a much bigger market altogether. So at the moment, we are approved as an exploratory biomarker. What we want is actual approval as almost a contrast agent. And as soon as we have that, not only can we expand how we're being used by pharma, so instead of just doing early clinical phase trials, we can be launched into their phase 3 trials and be used as a companion diagnostic for them, but also clinicians can use this in clinic. And that means we can help really streamline the way patients can be monitored. I suppose you would call it a personalized medicine approach. So every patient will have a risk profile and a treatment profile that will be governed by what DARC tells them. And these are the figures that and it doesn't look very conservative, but I can assure you it is, because we're only talking here about macular atrophy. But the market is big, the population is expanding, and the drugs that are being developed in this disease are going just beyond two, which came in through 2 years ago or last year, actually, to many others. So this obviously is due to a lot of people working to bring this all out to clinic. I'm very much helped by Alex from our BD department and Paul Finance. We've got a very strong science team, and this is a scientific type of company. I would call it a deep tech company because it's based on so much science. But we also have a very important board who help us a lot. It's very small. They're just 3 people on our board. Stephen represents our seed investment fund. Chris is also from another seed investment fund, and Natalie's an AI expert. But also very importantly, and I think just because I have been a glaucoma KOL for many years, it's been very useful in establishing KOLs around the world who have dictated, and we've pivoted. Initially, because my interest was in glaucoma, we were going to do glaucoma. But there is a lot more of a need at the moment with drugs that are available coming through in macular degeneration. And this is very much being dictated by the suggestions of our KOLs. I've included one of our investors from America here because Excite Ventures are an ophthalmic VC. Therefore, they specialize in ophthalmology. And it's very useful, bearing in mind at the moment we're thinking of going to market in America. These are the main investors that we've had to date. So we're moving forward now to the next stage. We would like to clear up, get our ducks in a row before we go out to Series A. I think the first thing we're keen on doing is sorting out the FDA regulatory pathway, whether they agree with us. We also want to make sure the reimbursement pathway and economics is correct. We actually have just got a grant for part of that from one of the Eureka schemes. And then finally, we are a startup, but we are negotiating with UCL Business about the licensing agreement. And then ultimately, we will move forward to our Series A, which we would hope to start next year. And that would allow us to go forward as a combination device in terms of the software as well.
Okay. Thank you. Thank you, Francesca. I wonder if I could sort of take you back to the spot count, as it were, in terms of DARC. I'm particularly interested in the convolutional neural network that you use in order to do that. And I noticed on your slide that you spoke about not only counts, you had things like density and some other measurements. And I happen to know that if I was building a model and I want it to be predictive in that manner, then I might want to sort of estimate density. And in the example that you showed of that poor patient who lost their sight, you said that you could predict, or if one had looked at it early enough, one could sort of predict what was going to happen. It looked pretty accurate from my point of view. If you could sort of unpack that a little bit more,
Yeah,
please.
sure. So it's not just one AI model. The simple model, which is the CNN, was basically the model that was used initially for identifying the spots. To be honest, that has even been improved now
Mm.
beyond the CNN to a more of a deep learning training type of algorithm, which is more accurate, more sensitive, more specific, and allows the morphology to be better. So the outline, the annotation of each spot to be better done. So we have changed with that. But like everything, you have to have a ground truth. When we did the CNN, the ground truth was 5 observers annotating the center of these
Hmm.
spots. And then the ground truth was where 2 or more agreed what the spot was. Subsequently, we then used 9 observers
Hmm.
and we'd increased that the agreement. But as I said, for that one, it was more of a deep learning rather than the CNN approach. We haven't published on that one actually, but it's our version 2, but it is definitely more accurate. And you, of all people, will know rubbish in, rubbish out.
Yes.
One of the things that we became aware of is the quality of the manual observer made a huge difference.
Hmm.
And what we've been most recently working on is to ensure that the annotators were trained and used to looking at this because no one's done this before.
Mm.
There's never been a system that has spots at the back of the eye that is used as a measure of any measure as of disease activity.
Okay.
So there is a little bit of sort of familiarity that you need to be sure that everyone is properly accounting for what has been positively labeled by our
Mm.
biologic. Um, so the stage it does evolve. So that's just the simple one. Then we go into the density map that I showed you, and that one is not too difficult because it literally is providing the type of geological geography map contour idea of spatial density.
Hmm.
The one we've been doing more recently, which which I would love to show you, but I just realized it still hasn't been published, is we're using a weather map idea, a polar plot idea,
Hmm.
or the prediction of direction. So you know, in a weather front, um, the way the wind can tell you which direction
Hmm.
uh,
Yes.
the front's moving. That's what we are using now for our atrophy analysis. And the reason for that is more based on science cellular level. So when I showed you that patch of atrophy, if you think of the retina as being consisting of different layers of film, film is the easiest way of putting it. It's one layer
Hmm.
of film on top of another. Um, in the atrophy that you saw, it's the bottommost film, which is called the retinal pigment epithelium, that has died. But above it, so has the next layer of film, which is the photoreceptors.
Right.
Now, what we have shown is DARC is predictive because before you get the bottom film destroyed, before the retinal pigment epithelium is destroyed, your photoreceptors have to die. And more importantly, more predictive of that is they have to be sick.
Right.
So our weather map application is looking at that front of photoreceptor stress as the warning signals as to where that patch is going to go towards the macula.
I see. I see. I mean, it's really, really interesting what you're doing. If I could sort of roll back, just to sort of be clear about, in sort of my parlance, what you have here. So, essentially, you're using a voting mechanism with humans in order to determine,
Yes,
yeah,
it is.
spots
Yeah,
as it were,
as
which
our
is fine.
ground
So
truth.
effectively, yeah, as your ground truth. So that would be in sort of AI machine learning parlance. That would be your training data,
It
and then
is.
you're going to yeah.
Yeah, indeed.
Um, and then you mentioned the deep learning model. I'm assuming that it is sort of neural net. It's a neural network
It is a neural
variant of
network, but
some yeah
exactly. I
description.
mean, it's cat. So initially, so maybe I should have said this before. Version 1 was template matching,
Right.
which was not the best. It was a thresholding, very rough.
Sure.
Wasn't the
A
best.
I'm familiar with deformable templates, so that was
Exactly.
that's a fairly classic pattern recognition method.
Exactly,
It's
which
been
is
around for
a
some
bit
time.
old-fashioned, I gather, but
Yes, so convolutional neural networks
yeah,
tended to overtake that.
yes,
And many people will regard CNNs as deep learning in themselves.
yes. Uh, but I mean, they are to many respects, but you still need your ground truth, don't you? I mean,
Correct.
we haven't given it we haven't given the black box a whole lot of data and say
Sure.
this and see what, because the point here is that the fluorescent signal needs to be taught.
Mm.
Yeah.
Yeah, no, I understand that. I mean, I was just thinking if there was a way to create synthetic data.
So
Uh,
we've thought about this as well because I mean there's good and bad because we use the biologic, and biologic is the only way of creating these dark images. Um, the good is that it's proprietary. The bad is that it's limited because
right.
only those people who are using our biologic and
Mm.
only us who have access to the images can basically use that in our database. It's not like, and this is where it's so different when people talk about AI in the eye, they
Mm.
talk about those OCT scans that your optician is regularly
Yes.
doing, yeah. And those are available and accessible from multiple sources. And basically, if you wanted to, DeepMind did an algorithm for that. Everybody's doing algorithms for that because there's lots of data, and
Mm.
it's big data, not only in terms of each scan has a lot of data within it, but there's a lot of people who have regular scans. We are limited by by the number of patients who've received DARC, and so it's a small data set, and we've been looking into synthetic data because of that.
Mm.
Um, there is, I mean, you will know this recent publication, was it last year, about they showed that synthetic data is good, but at some point, if you feed synthetic data as your training data, it spews out rubbish.
Sure. Sure. Certainly in a large language model context, that absolutely.
Yeah,
But it is used. I mean, if we're using a technique known as reinforcement learning, then a reinforcement learning model can create its own data, but that's a very stylized environment that it operates in. Um,
but it's something that I have three of the team are data scientists, in fact four, and they've looked at that. But I wouldn't say, to be honest, each time, every time you begin using something, it quickly becomes out of date in in AI terms, doesn't it? And it's certainly something that we've been finding. But the synthetic and reinforcement is something that my guys have been talking about. I mean, I would love it if we could do um, improve with synthetic data, but I don't know
hmm,
how much of improvement we get based on the fact we've just got this small data set. It's 400 patients, but they have multiple images.
yes, yes, sure. Um, yeah, I mean the worry with sort of the generation of synthetic data, I mean it's almost like the bootstrap, so sampling with replacement from the original data set is that you really need that sample to be sort of representative of the population as a whole. If it isn't, there's an awful
Yeah,
lot that's
then
going
you
to
could
be
be
missed.
yeah, yeah, and absolutely, we don't want to be accused of overfitting. And actually, if you look at publications from the world of eyes in
Hmm.
AI, that was one of the biggest issues before. And also, I think what we've suffered from in the past, if you people don't publish positive predictor value,
Yeah,
which would be the real test of things. But yeah. Talking of which, we've had pretty good results with our PPV, but I can,
right.
depending on where we set the cutoff, for example, in our paper with glaucoma, if we set the cutoff at 30, all patients who had a dark count greater than 30 went on to progress 18 months later.
Right. Yeah. I mean, yeah. I mean, it's unfortunate about the progression, of course, but the diagnostic power is hugely positive.
Yes, it,
Yeah.
it is. It is. So, I mean, it'll be a paradigm shift, Richard. This is quite interesting because it will ultimately replace what we are routinely using in
Mm.
ophthalmology and potentially even in neurology, um, in a way that you wouldn't have thought possible. And you know, it's exciting, but it could be thought of as being so disruptive that it's doing something different and risky.
Okay. Okay. Well, that sort of brings me nicely on to where I was first intrigued by your research, actually, as a diagnostic tool for these neurodegenerative diseases, particularly Alzheimer's. I mean, Alzheimer's is, well, it's an iceberg that's facing the world, but we sort of stick close to home. The NHS. This is I mean, it's huge from any way you look at it, but particularly from a cost perspective. So anything that gives you a early stage indication has got to be hugely beneficial. And I happen to know that actually, if you are going to treat Alzheimer's, the earlier the better, even with the things that we've got to hand now.
Exactly, exactly. And that's true for any of these neurodegenerative diseases, including those in the eye. Your chances of modifying and stopping the person progressing, your patient progressing, is much greater the earlier you identify. And in fact, if you look at the Alzheimer drug trials, when they managed to stratify the patients according to what stage they were when they were started treatment, it showed that exactly. It showed that those patients who had early disease had a much better endpoint than outcome than those who had advanced disease at the time the treatment was started. And that's true for a lot of these uh, diseases, indeed. So in terms of neurodegeneration and why the eye could be used. And it's something I've been talking about for many years. And I don't know if this is true, but some people think that I coined the expression that the eye is a window onto the brain. Because in terms of the fact embryologically, the same cells that form the eye also form the brain.
Mm.
The parallels of what's going on in the brain can be seen in the eye. But because the retina is more accessible, you don't need an MRI, CT, or a PET scanner. You just need to image. It could potentially be much more powerful as a screening tool. Um, so we've had some evidence of this. A lot of it has been preclinical, showing that we can pick up changes in the retina before you have the changes in the brain.
Mm.
In particular, for example, beta amyloid and tau deposition in the brain. We have shown that we can pick up DARC positivity in the retina in transgenic models well before that occurs. Um, we have also been, I mean, in our preclinical Parkinson's models, very similar thing. And also in those models, we've also shown that we can use the eye to monitor the treatment. So even though it's a treatment in the brain,
Hmm.
because the eye also has amacrine cells, which are part of the dopaminergic system, um, a good treatment reduces the dark signal because you don't have those cells in stress.
Right.
And also, there is more going back to our OCT AI colleagues. There is more evidence even through OCT that you can potentially also use the OCT as a marker of those diseases. I'm not sure how specific or sensitive it is for those diseases, but it is just adding validation to the fact that changes in the eye are indicative of those that are going on in the brain.
Right, right, right. And of course, this is why you've partnered with these pharma companies because of course they want to,
Absolutely,
yeah.
but actually, Michael J. Fox, we've got a grant with at the moment for looking at dark with patients with Parkinson's.
Mm.
And we had a grant from Innovate UK for looking at
Hmm.
patients with multiple sclerosis. We haven't really concentrated on Alzheimer's, though I would love to. But the issue is with Alzheimer's is it's been a bit of a it's not been a smooth run for pharma.
Right.
And I think it's more difficult to get funding, therefore, because of the fact that it's a more difficult area to break into.
Oh, I see. Right.
I think that would be, and until a very convincing drug comes through, uh,
Yeah.
I think so that I mean that's my take on it at the moment. I may
Okay.
be wrong, and I'm very happy. I'm very open to other people's opinions on what
Mm,
they think
mm.
that should happen.
Well, I mean, my take on it was the fact that you've got an early stage detection
Yes,
mechanism, and
exactly.
that's got to be, potentially of huge value. Um, that's why I was coming at it because, we do have some drugs that can be used to delay the onset of Alzheimer's. There's no
Exactly.
cure.
Yep.
Um,
As long as it's detected early enough.
so why hadn't I heard of Novai before we met? That was sort of my
Because we
my
haven't
question.
gone out saying that we are doing neurodegeneration
Yeah,
for Alzheimer's, I think would be fair to say.
right.
Partly because the most of the evidence that we've accumulated in patients is for eye disease.
Correct. Oh, correct. Yes, I get it. Yeah,
Yeah.
yeah. So I mean, this is very much a sort of a potential spin-off benefit.
Yes, indeed. I mean, if there was funding from somebody who specifically wanted us to concentrate on Alzheimer's, we
Hmm.
would. We really
Right,
would. Yeah.
right.
Um,
That's it.
I would like to have done everything yesterday, Richard, but
Right.
unfortunately, you need money.
Yeah, yeah. I understand. I understand. Well, on that sort of topic, I mean, you describe yourself as a startup, but I have to say that Novi strikes me as being, it's very much a mature startup. Certainly in terms of, what you've done so far and the amount of funding that you've received. But obviously you have great ambitions here. So where do you see that? What next? Where do you see the sort of path of travel in terms of the business, as it were?
So, I mean, it's an interesting side of things that you've mentioned. You are right. Compared to a, suppose we were a therapeutic, the amount of evidence that we've accumulated would be considered almost late stage because
Mm,
we were a therapeutic. The issue is because at the moment we are a exploratory biomarker, and even though exactly the same technology is being used across the board, going from clinical trials where we're being used to test drugs to being integrated into clinic is seen as a big step. Now, I'm not sure whether the messaging is confusing because actually we've already proven the fact that A, we can stratify patients, and B, we can look at treatment responses. But it seems to be um, it's not another product, it's the same product. So I mean, I
yeah.
don't know. I think maybe and Richard, with your worldly wise hat on, maybe you could help, what would you have thought? Because I my gut reaction is agree with you that I think we do risk a lot of things in our company, and we're obviously revenue generating as well. Um, how do we ensure people understand that we're already validated, as it were?
Well, what I find is that in terms of sort of fundraising and interest, the messaging needs to be clear, and I think certainly this seminar has really helped me. Um, but people tend to be quite sort of linear in their thinking. They want to put you in a particular box. And once they've done that, it's very hard for them to sort of think outside that box. And I think you probably suffer a little bit from that. So, you need an investor who has a bit more of a creative thinking mindset. But of course, the large investors, they employ, individuals who have got a very specific sort of remit in terms of the information gathering that goes back to the, mothership, as it were. I think, I mean, talking to more people,
Yeah,
um,
yeah,
uh, you know, uh, presenting Novi in as many places as possible, diverse pockets, as it were. I mean, you mentioned Michael J. Fox. Obviously has a personal interest, but
yeah, yeah, yeah.
Michael J. Fox wouldn't necessarily be a first port of call for many people in this area, as it were. So things like that, I think, are good. Just getting people interested. I mean, it sparked my interest really from that neurodegenerative point of view, as opposed to the eye technology directly.
Sure.
Um,
Yeah.
and also, I
AI
like
is
that.
still
Yeah.
quite niche, isn't it? In terms of uh, unmet needs as well. Uh, Neurodegeneration is fatal. Vision
Yes.
loss is very
Yes.
important. Yeah,
Yeah.
uh, absolutely. Yeah, no, I get that.
Mm. No, I don't want to. I don't want to be negative at all, but I do want to sort of touch on the challenges that you faced, particularly in terms because you speak about sort of clinical approval, etc.
Yeah.
So navigating all that. If you want to sort of touch on that, and you're looking at FDA approval in due course.
Yeah, so I don't consider that a huge challenge um because we've already been used a lot, and every time we are used by a drug company in a trial, we go through another assessment from the regulatory authorities, and we
Hmm.
always get back. We've even got a drug master file with the FDA because that allows people to look at all our data in a secure environment, but
Mm.
also shows that the FDA obviously have already assessed us. If it was felt that there was any type of problem with our safety and tolerability, we would have known by now. The
Yeah.
regulatory authorities would have cracked down. So it's not a challenge as such. It's more to do with the fact that we have to formally apply
Hmm.
and make sure that the package is consistent with whatever the FDA requires. So I don't think that's the challenge as much. I think more of a challenge is more up your alley, which is the lower bar is the biologic getting approved. The
Hmm.
higher bar is the device. So the
Right.
software, along with the biologic, it's a combination device. That is more of a challenge because in terms of software approval, software as a medical device
Yeah.
approval, the a number of images or validation you need has historically been huge. I say historically because very recently there was a company that the FDA approved um, their AI use for an at-home device with not a huge amount, not hundreds and thousands or even thousands of data points or images.
Mm.
So when we talk about the Series A, what we've really been trying to use the Series A is to get more data, going back to your question on synthetic data, to get more data that will validate the software for the FDA to approve it as a device.
I see. Yes, yes, of course. So the wonderful thing about software, of course, is it's the one product that one can release that isn't necessarily finished.
Yeah,
Or rather, when something better comes along, provided the software can be updated,
yes,
one can do that.
yes.
So I mean that's my recommendation for any device, but I don't,
No, no,
don't
that that
know.
that's very true. And actually, I mean, just so that you're aware, um, a lot of in the eye world, there are some AI software algorithm and companies that have been used as investigational devices whilst they gather their data to get validation as an approved. And that is really what we would be planning to do is to be used as investigational. And because we're going for a clinical decision support tool, so we're not actually giving the clinician taking the power of the clinician away. We're saying this is what we would suggest you consider when you decide on whether your patient needs more treatment. So it's a support. Um, it's less stringent than if we were to say it's being used to say whether you have disease, yes, or whether you need treatment, yes. There's no black and white rule.
Right, I see. Okay. Just some, I've just got some sort of personal questions uh, to sort of round up with. What's been the most rewarding aspect of seeing DARC from sort of concept to significant clinical potential?
So I think a lot of clinicians, a lot of scientists never have the opportunity of actually seeing what they've been working on being used in patients and getting to clinic. And I think that remains for me the most important reason of why I still do what I do. I'm working not quite 24/7, but I'm working pretty hard um, to realize this, to allow it to get
Mm,
further. And I see my mission is to try and make this accessible to as many people as possible. And I shouldn't say this, but I've never been in it for the money. I've really been in
mm,
it to try and help. And it came from a personal experience, actually. So it's as a clinician, the worst possible experience you have is you have a patient who's losing vision coming to clinic regularly. You've thrown everything at them.
mm,
You've thrown drugs, you've thrown laser, you've thrown surgery, and they're still losing vision. And that is really what spurred me on into getting into technology that could identify much earlier when that patient had the disease, or when, in other words, when they could be treated earlier and therefore prevent them losing that vision. So
mm,
going back to what you were saying about Alzheimer's, it's exactly the same in vision loss as well for these diseases.
mm, mm. Yes. Um, yeah, I mean, I don't know how you balance your time. You say you're working 24/7. I can well believe that. Um, so just sort of jumping ahead, might we see, and this is a little bit of a tongue-in-cheek question, of course, but might we see a stock market listing at some point for Novai?
It would be nice, as you know, there's no one's been particularly successful at an IPO in from the UK in several years. I think Raspberry Pi probably
Right.
might be one of the few big ones that did. And everyone, is saying don't put an IPO as your first exit.
Mm.
Perhaps we should be putting acquisition. And we
Hmm.
know we have potential partners who'd be very happy to do that. Personally, I don't know. It's just because it's the passion, and what I see is my mission in life. But I'd like to take this as far possible so that its utility can be realized. And I don't mind how we get there, but I'd love to be part of that journey.
Right. Fantastic. So, look, I mean, Professor Cordeiro, thank you very much for your time. It was fascinating tour of Novi and the technology behind it. Um, and yes, thank you very much.
Thank you very much. It's been a pleasure talking to you. Thanks a lot, Richard.
Bye bye.
Bye bye.
If you enjoyed this seminar, you might like to take a look at our istonline.org.uk website. You can find recordings of many of our previous AI seminars on our IST Online YouTube channel. Thank you and goodbye.